Well, welcome everybody. Welcome to our second day of the Almond Conference. Happy to see you here. We have just 1 hour for this session, and it's an hour where we're going to try to summarize a lot of research. This session is called Production Research Updates. So, as the name says, we have aligned different research projects that we have supported for over the years. I have asked the lead researchers of those projects to give us a 10 to 12 minutes update of what they are working. And that's a big challenge for them because they have collected a lot of data. They have been working for multiple years. So thank you to all the speakers here for summarizing their years of discovery. I'm Sebastian Saa. I'm the Associate Director of Agricultural Research at the Almond Board, and I had the pleasure to work with them and with many more researchers that are also in the poster session today downstairs. Today is the day that we lay out all of our research programs. So if you don't hear what you are interested here, it's probably going to be down in the first floor as well. So a double opportunity to catch up with what we're doing today and what can shape our future for tomorrow. So I'm going to introduce all the speakers at once, and then I want to really let them take over. So the first speaker here, we have Andreas Westphal from the University of California, Riverside. He has been working a lot in nematodes and how to control nematodes. I still don't know if they're diseases or insects, but somewhere there I know that they are really challenging to control. So thank you, Andreas, for your work there. Then we're going to move to Mohammad Yaghmour, also from the University of California, but from the ANR, from the Farm Advisors system. He has been working in whole rot, how to control whole rot, and he's going to give us an update along those lines. We'll then move to Isaya Kisekka. He's a UC Davis faculty there, and Isaya has been looking into irrigation 2.0, meaning like a little bit of a smart irrigation and the technology that is around and how we can move forward with that. We'll then move to Roger Duncan, or emeritus farm advisor, also from UCNR. Roger has spent his whole professional life working in almonds pretty much, and he has one of his rootstock trials is evaluating rootstock compatibility and options for off-ground harvest. So he's going to give us an update of that project as well. And then last but not least, we're going to cover Franz Niederholzer over there. Franz is in charge of our strategic collaboration with Nickel Soil Lab. Nickel Soil Lab is a farm, a commercially managed farm that is in Arbaco, California, a little bit in the north of the valley, and has been really a hub for innovation and research and extension. We have supported small projects there and big projects like long-term projects like organic management for 20 years of data and so on. Franz has been a great collaboration, and he's going to try to summarize most of that. With that said, I'm going to pass it to the speakers.
Good morning, everyone. My name is Mohammad Yaghmour, and I'm the Orchard Systems down in Kern County. And today I'm just going to speak about some of the work that we have done over the past years with the generous support of the Almond Board here about whole rot of almond disease, with the focus of Aspergillus niger as the causal agent of the disease. So as some of you know, there is 3 important parts of the disease triangle. The first one is we need a susceptible host, and we need to understand which pathogen is causing the disease and the sources of inoculum for that pathogen. As well, we need to have the conducive environmental conditions that will lead to the formation of the disease and the problem. So whole rot disease starts with the infection of the hull with one of the causal agents. As the disease progresses, what really we see, as you can see down in the corner, the leaves around the infected fruit will become dry, right? And the whole spur that is bearing next year's fruit will also die and also may cause the death of whole shoots, not only the spur. So we did go ahead and we quantify the yield reduction of next year, because what happens is that the disease takes place this year, and it will kill the spurs that will bear the fruit for next year. So as you can see here, we did that over 3 orchards, where actually we saw that the reduction really mainly happens when we account for 100 strikes on those trees or more, we start seeing reduction in yield. So we have 3 major causal agents. The first one to the right on my screen here is Monalinia species. We know that Monalinia can cause hair rot. It's the same pathogen that causes brown rot during bloom. So how do we know that it's the pathogen that we are dealing with? You can see the signs, which is usually the spores on the outside that they are tan to a little bit of brownish there. The other one to the far left is the Rhizopus stolonifer, which was always the culprit in the Central Valley. You can see that the signs of it are fuzzy growth of the fungus, and the spore, they look more of gray spores. Sometimes they look bluish under the sun. However, in the past few years, since 2017, we have seen an increase in the Aspergillus niger, which looks very dark to jet black spores and flat in nature, not as fuzzy as the Rhizopus on the left. So again, as I mentioned, we have start seeing that especially in the warmer areas, because this fungus thrives in warmer temperatures. We start seeing it a lot in Kern County, and after a few years, we start getting many reports of it throughout the Central Valley. So what happens, you can see the difference sometimes also in the symptoms, where Rhizopus to the right, beside you can see the signs of the pathogen and the spores. If you don't have the fruit, the infected fruit there, and you just look at the wood, you will see the black streaking. That's due to the production of the fumaric acid, which we believe is the causing of the death of the shoots and the spurs. On the left is also showing symptoms of Aspergillus. You can see that the peduncle and the spur actually are dead and brown compared to the green live tissue of the shoot around it. It's not always when you look at the orchard and collect samples, you're going to see one or the other pathogen. So down south, when we mainly collected quite a bit of fruits from infected spurs, we quite a bit saw Aspergillus. We saw sometimes Rhizopus alone, okay, with high incidence of Aspergillus. However, sometimes you get mixed infection, where you can find both of the pathogens together. And later on, you will know why it's important to ID the pathogen, because Monalinia, the way we treat Monalinia is totally different from the way we're going to treat Rhizopus and Aspergillus, and that is mainly due to the source of inoculum. This is just to prove, that the soil is the source of inoculum for Aspergillus flavus, as well as the Rhizopus. So if you can see down on the charts, what we did is simply collected some fruits, wash it with water, and plate it on plate to see the population of Aspergillus niger in the soil. And you can see exactly the peak of those spores around the end of July, early August, when actually the hull split takes place. Some of that results also is a result of the fungus start to grow also inside the hull and produce new spores. So it's also important, beside knowing the sources of inoculum, to know what is the most susceptible stage on the plant side. Dr. Jim Scavage did great work before, where they know that the deep V stage, you see it in the picture there, is the most susceptible stage for Rhizopus infection. However, we didn't know exactly what is the most susceptible stage for Aspergillus. And that's when we did some of the work where actually we inoculated some of those fruits at the different stages. You see that, while you get some significant infection at the deep V, most of the infection and the high severity happened when we have the hull split less than 1 centimeter, which is, if you look here, oh, it's not going back. Oh, here it is. It is the C1 less than 3/8 of an inch. That was the most susceptible one. So knowing what we know also, it's important to know also what is the most susceptible varieties, because that's also will take in account the way we are going to manage this disease. You can see some of the most susceptible varieties. This work was done with David Dahl and Brent Holtz. Nonpareil, Butte, Sonora, they are one of the most susceptible ones. We did also some work that also coincided with their results on the graphs on the right. In the same orchard where we had 3 varieties, Nonpareil, Fritz, and Monterey. And you can see that, Monterey is not as susceptible as Nonpareil. Similarly, Fritz. And we can have quite a bit of orchards that have those 3 varieties down in Kern County. So when it comes to management, we have to take good care of the nonpareils because we are expecting that nonpareil will be more severely affected by the disease compared to Fritz and Monterey. What are the strategies that are available for us to manage this disease? We have many strategies, but we always focus on the protection strategy when it comes to IPM strategies. And that will include mainly for this host resistance, cultural practices, and chemical control. When we are talking about host resistance, we don't say that some varieties are more resistant to others, but they are less susceptible, as you have seen before. When we are talking about cultural practices, which is an important part when we are dealing with whole rot management, I'm going to talk first about the management. I'm not going to expand too much about the management of nitrogen, but over-fertilization with nitrogen has been proven before, and there's more than one paper out there that shows that if we are over-fertilizing with nitrogen, we're going to have more disease with Aspergillus niger and with Rhizopus stolonifer, and the use also of strategic deficit irrigation decrease the disease caused by Rhizopus a lot. Fungicide, they work, but they are not the only solution. So when we are looking at the fungicide work, I did some work with the fungicides, and we know that from the table of the most effective management practices that, during the hull split time, sprays are the most effective ones. That's why I'm saying we have to know exactly what we are dealing with. If we are dealing with Monalinia, we have to spray the trees 3 to 4 weeks before the hull split. But if we are dealing with Aspergillus and Rhizopus, then the hull split sprays are important, and usually that coincides with the navel orange worm. We did some work for in vitro showing that frag group 7 and 11 and 3 works very well. I did that also in the field, and here you can see at the 2 different stages, the most susceptible one showing that, at hull split, less than 3/8 of an inch is very effective. So moving on, this is my last slide that will summarize everything. So that's why it's important to know exactly what we are dealing with. And a good example is if we are dealing with Monalinia, we have to spray 3 to 4 weeks before. If we are dealing with Rhizopus and Aspergillus, then it's at hull split. Irrigation management and avoid excess nitrogen is important. There is great resources from the Almond Board and the UC regarding that you can find on their website. Dust management is really important during hull split, because the source of inoculum is in the soil. If you're creating dust, you are actually moving the spores from the soil to the canopy where they can infect the susceptible tissue. And I mentioned the chemical control very well in a way that when we are dealing with Rhizopus and Aspergillus, it's during hull split between 1% and 5%, whereas the Monalinia is 3 to 4 weeks before hull split. That's my last slide. Thank you very much. And if you have any questions, please feel free to reach me. Thank you.
My name is Isaya Kisekka, and I'm a professor of irrigation and water management at UC Davis, and I'm going to talk about data-driven precision irrigation. So one may ask, why do we need to do precision irrigation? There are many reasons. One is the chronic water scarcity due to repeated drought cycles. If you look at this graph, it shows the number of droughts we have had in California over the last several years, but you see the intensity and duration of these droughts has increased over the last 20 years. And on top of these climate extremes, you have sigma. We now have pumping restrictions on groundwater. Growers also have to deal with raising input costs of energy and labor and fertilizers, as well as fluctuations in prices. And so one of the tools that growers could use to cope with these challenges is precision irrigation. And so what is precision irrigation? It's the idea that you're applying the right amount of water in the right place at the right time. It's not just about the right amount of water and the right time. So on this slide, you're seeing an orchard. This is a 15-acre orchard at Nico's Soil Lab, and the different colors show stem water potential expressed as bars below baseline. So you can see that parts of the orchard are experiencing water stress, and other trees are actually happy they don't need as much water. But if your irrigation system is plumbed in a way that you can only apply the same amount of water, you'll put on the same amount of water everywhere. And that would mean you're overwatering some areas and you're underwatering some areas. So precision irrigation can help you deal with this problem. So in our Almond Board funded project, we wanted to study two ways to implement precision irrigation. One where we irrigate by variety, and the other where we break the orchard into different zones that account for differences in soil infiltration, soil water holding capacity, and other factors. So this is the orchard that I showed in the previous slide. It had three varieties. It had Aldrich, Nonpareil, and Butte. We reprogrammed the system so that we had the ability to irrigate each of these varieties independently, so we could maximize the yield per unit of water input. So the way we did this, we had two strategies. In the first strategy, we implemented regulated deficit irrigation when the Nonpareil variety reached whole split. So we reduced irrigation by 25% and then 50%. And in the next strategy, we implement this regulated deficit when each independent variety reached its whole split. And so what we found was that there was no significant differences in yield and nut quality. And part of the reason was this was a 15-year-old orchard, so the strategies we were implementing in 2 or 3 years did not seem to have a significant impact on yield. But going forward, we are repeating this study in a young orchard to observe the differences in yield over a longer period of time. If you're interested in the details, they are all in this published paper. The next strategy was to understand how differences in soil variability impact yield. If you look at a satellite image or an airplane image of an orchard, it's very rare to find an orchard that is uniform everywhere, and most of the variability is actually driven by soil. So what you're looking at in this particular orchard, this is an orchard near Corning, California. The red areas or orange areas are actually heavy soil, heavy clay soil. The green areas are layered soil. You have heavy soil on top, and then you have light soils on the bottom. And the blue areas, you have sand and more gravelly soils. And so what the grower did to address these challenges, he broke this orchard into smaller zones. Because if you walked into the orchard, some areas would be flooded because the soil intake was so slow, and other areas, the intake was very good. So they had three zones: the red, the green, and the orange. The red was the light soil, the green was the layered soil, and the blue was the heavy clay soils. And so we wanted to understand how these different soils actually affected irrigation. So we used a technique that's widely used in oil and gas industry that allows you to look in the subsurface, where you inject electricity in the soil and you use it to tell you where water is. So I'll start by showing you a few slides on the heavy soil. So what you're looking at here is if it is pink, that means the soil is drying, and if it's blue, the soil is wetting. And in the heavy soil, you can see the pink, which shows root water uptake. So root water uptake is occurring at a rate that is faster than the irrigation. And this is why you tend to see a lot of variability in clay soils, because the root water uptake and infiltration are not very uniform, which is different. If you look at a light soil, for example, the infiltration rate, which is blue, is occurring at a faster rate than root water uptake. That's why we have more blue than pink. And actually, this is reflected in the orchard. If you look at the orchard, the one in the light soils tends to be more uniform than in a heavy clay soil. So what does this mean for a grower? If you have heavy soil, it's better to have less frequent irrigation. And if you have light soil, it's better to have more frequent, smaller irrigations. In terms of yield, these are the yields in the different irrigation zones. The light blue is the yield, and the pink is the amount of water applied. You can see that overall, the light soils actually had better yield than the layered soil and the heavy soil. And this is actually has been observed for a longer period of time. In clay soils are much easier to compact. Root distribution also tends to be more restricted, and you also can have aeration problems. And so we computed water use efficiency, and although the clay soils had lower yield, they had the highest water use efficiency because they required less irrigation. And water use efficiency is just the total net yields divided by how many inches of irrigation you applied. So we compared this with what my colleague David Dole computed a few years ago when he took data from different sites. He got a water use efficiency of 61 pounds per inch, and we were able to get much higher water use efficiency in the different zones. So how were we determining when to irrigate? We had three strategies. We monitored soil water, we monitored stem water potential, and we monitored ET in the different zones to determine when to trigger irrigation. This is an example of a soil moisture sensor, and this can help us in two ways. It can tell us when we are leaching. If we see the purple line at the bottom, which is at 48 inches, start to increase, that means we are leaching nutrients or irrigation. And you can see that whenever we irrigated, which is the cyan bars, you can see that the top two sensors at 4 inches and 12 inches were responding to the irrigation. The other thing that we experimented with are these sensors that you embed in the trunk of the tree to tell you if the tree needs irrigation. The good news is they worked better in almonds compared to other tree crops. And the orange line is the sensor, and the black lines is a pressure bomb. Many growers don't have time to go with a pressure bomb to take measurements, but with these types of tools, they can give you continuous data that is actually pretty accurate. We also studied low-cost sensors that measure how much water is going off an orchard. If you're in a GSA that has water allocation, this could be one of the tools that you can use to say, okay, this is the actual amount of water that my orchard used. And you can see that the agreement between this low-cost sensor and some of the very expensive scientific tools is quite good. We also developed an app. If you're interested in playing with it, it estimates attainable yield, not actual yield, attainable yield as a function of the size of the canopy. And it builds on the work of my colleague, Bruce Lampenin. It only works on iOS devices, not Android yet, but we're working on making it work on Android as well. So you just take a picture and it will give you estimates of attainable yield and what irrigation you could potentially apply. So in terms of what this means to you, these methods are scalable. They can almost be implemented on any orchard. And they have benefits in increasing how much yields you can get per inch of irrigation water. I'm running out of time, but the take-home message is there's potential with these technologies to improve water use efficiency. I'd like to acknowledge the Almond Board and the USDA for supporting our research. Thank you.
There we go. Good morning, everybody. I'm Roger Duncan, and today I'm going to talk about kind of a fun project, which was looking at size controlling or dwarfing rootstocks for almond production in California. It's very different than what we're used to, especially in the San Joaquin Valley, where we've kind of been moving towards an even more vigorous rootstocks. But this is a photograph that I took when I was in Spain in 2001. I cannot believe it's been 24 years already, but so this is a super high density system where they're using a very dwarfing rootstock, and you can see the idea here is to make a kind of a fruiting wall, and at that time the spacing was about 12.5 feet between the rows, and my understanding is that now it's, I think, closer to about 10 feet between the rows, and the tree spacing down the row is still right about 4 feet. So at that time, it was about 900 trees per acre, and now it's over 1,000 trees per acre in that system. And it has become pretty popular. Here's a field day. You look at the Spanish growers looking at this system. And what I noticed, though, is there's a lot of land that is dedicated to driving space and not necessarily tree space. But the idea here is that the trees are more efficient than in our sort of larger tree system. There are other systems, though, that can utilize off-ground harvesting, and this is a Tinayas harvester, which is also out of Spain, but this allows the trees, or this system allows for larger trees. So a maximum tree height of about 18 feet and about 18 feet also between the rows. So fewer trees per acre, still an off-ground system. And in Spain, off-ground makes a lot of sense because the ground, at least from what I saw, was much more uneven, a lot rockier, and so it's much more difficult to harvest off the ground. I think I'll skip over that. And there are also other off-ground systems. Here is a toll system, for instance, and there are others as well. But this machinery doesn't necessarily require a smaller tree. So here's a kind of a drone photograph of the trial. It's at the University of California Kearney Ag Center down just south of Fresno. And you can see there are groups of 10 trees, nonpareil in Monterey. And so you can just see there's a lot of different tree sizes in this plot. And the idea of this was not necessarily to look at this high density system, but really more of a cataloging of these rootstocks that we have in California now. And if somebody wanted to start, to look at using a dwarfing rootstock, which ones do we have now? How are they compatible with almond? And how dwarfing are they relative to a standard such as Nemigard, for instance? So here's a list of the rootstocks that are in the trial. The upper part of that list are replicated rootstocks. So each rootstock is planted in one location, and then another location, then another location, and the averages are taken so we can have a better confidence, I guess, in the results. The bottom rootstocks are ones that either we couldn't get very many trees on those rootstocks, or we had to wait a year. So those are really more for observation and not necessarily replicated data. But you can see there's quite a bit of number of rootstocks from various nurseries: Zager Genetics, Brights, Agromaria, and Davis, Wawona, USDA, Fowler Nursery. So quite a bit of buy-in. Most of these are numbered rootstocks and not into production as of yet. So the trial kind of looked like this, planned in the fall of 2020, and the orchard was planted thinking that maybe we would harvest it with a Tinayas system, or at least planted at the distance that would accommodate one of these Tinayas harvesters. So not really a super high density system, but certainly a high density system. Planted 8 feet between the trees down the row, and the row spacing was 18 feet between the rows. And instead of staking each individual tree, we used a wire trellis. So a single wire trellis, kind of like a grape trellis. And the idea also, no scaffolds could be allowed below 3 feet. That's what some of these harvesters require. And which was handy. You could just take everything off below 3 feet, and there was essentially no training or no pruning above 3 feet. And here is the system in the spring. Everything was cut back to, it's just a single, like a dormant tree. And you can see kind of the end posts here in the wire trellis system. And each tree was held up by one of these clips. So fast forward 4 years, this is Nemagard, which has now really pretty much filled its space in that allotted time. And it also becomes pretty clear that, this is at 4 feet. Very soon, this is going to outgrow its space. So it's already reaching across the aisle there at 18 feet. And so this is why we typically don't plant Nemagard at the rows 18 feet apart, because it's starting to really fill up quickly. You can compare this to the most dwarfing rootstock in the trial. This is root pack 20. This is the one that is typically used in Spain, and it was the most dwarfing rootstock in this trial. And you can just see the difference in the tree size and the canopy cover there. So I'll go through a few rootstocks, just some of these you may not have heard of, but just to kind of run through some of the results here. So this is root pack 20. And we noticed the first 2 years, there were quite a few signs of what I would call incompatibility or minor signs of incompatibility. So this leaf rolling, the growth stopped around, early in the year, leaf rolling, leaf burning. And then on some trees, if you cut right at the union, you could see a little bit of a line that indicates either some starch buildup, not a real complete union there. But after about year 2, those symptoms went away. And so the trees have grown well since then. However, the suckering problem that you can see on the right there, that continues today. That has been, I have to say, a little bit of a hassle with this rootstock, quite a bit of suckering. Controller 9, which used to be called P3135, this is a peach rootstock, and it's called Controller 9 because the vigor is supposed to be about 90% of Nemagard. With almonds, or at least in this trial, it is definitely not 90%. It's much, much less than that. And in fact, both Monterey and Nonpareil showed extensive signs of incompatibility. A lot of trees have died, well, half the Nonpareil have died. And again, we saw a lot of this brown right at the union. So I would say that Controller 9 is probably, I would consider just to be not compatible with, well, at least Nonpareil and Monterey varieties. And there are other varieties that showed incompatibility with one variety and not another. So this is DA6, which is from Sierra Gold. It has now been named Bantam. This is a rootstock for plums. But again, trying it on almonds just to see how it looks, how it works. And what's interesting is the Nonpareil trees failed miserably. But the Monterey trees, they look pretty good. And you look at them today, well, not today, but you look at them a couple of months ago, and they actually looked really pretty good, and it was, fairly good sized trees, at least for the goal of this trial. So real difference in compatibility between or among the varieties. And here is citation. This is an old rootstock that is used quite a bit in stone fruit production. We had the opposite effect. The Nonpareil trees look really pretty good, and the Monterey trees just struggled, and a lot of them have died. So this incompatibility thing is really interesting, and it's not black and white. It's all kinds of shades of gray between compatible and not compatible. And then, of course, the varieties are very, very specific as well. So all of these would have to be worked out before anybody decided, I'm going to go plant an orchard on such and such rootstock. You got to make sure. It's compatible. MP29 rootstock out of the southeast, brand new, is bred for our malaria resistance. As far as I know, this is the first time it's been tried in California. Seems to be compatible with Nonpareil and Monterey, although you can see the growth is it's a little bit sparse. You kind of see the plum effect, that Nonpareil kind of more straggly. But the trees look pretty good. Controller 6. This is one of the better looking rootstocks. It started out pretty size controlling, but now actually the trees have grown to almost the size of Nimegard. So I'm not sure I would call this a dwarfing rootstock anymore. It was for the first couple of years, but now they've pretty much almost caught up to nonpareil. Okay, so I know there are a lot of numbers here, and all of this is on the poster, but just looking at the top rows, these are rootstocks that are in red I would consider to be mostly incompatible with almond, or at least certainly with one of those two varieties. Citation is pretty iffy, and then the rest of the rootstocks, we were not seeing any signs of incompatibility. Trunk circumference, we have a lot of other measurements too. We did some scan the orchard, so we have tree height, we have par measurements, leaf canopy area, all that kind of a thing, just hasn't been finalized yet. But you can see there are a wide range of trunk sizes or of dwarfing among these rootstocks. And then finally, we looked at yield. This is something that you really have to take with a grain of salt, because obviously all of these trees are various sizes and everything is planted at 8 by 18. And I think that the conventional rootstocks, Nimegard, Brights 5, and Viking obviously fill that space very quickly, and we got higher yields much sooner. Even fifth leaf, much higher than all the rest. The rootstocks on the bottom, well, a lot of those were incompatible or had a lot of incompatibility. Root pack 20, cumulative yield is only 1,500 pounds for third, fourth, and fifth leaf, so not very high. And it's just, again, this is a small tree, and 8 by 18 it's just too wide of a spacing. So this is not a knock on root pack 20. It just needs to be planted much more densely than at this spacing. So again, I'm not saying that these rootstocks are good or bad. I think it's just, the spacing is obviously needs to be tailored to the vigor of the tree. And I think that's it. So thank you for listening. And I'll see you at the poster if you have questions.
Good morning. There you go. I'm Franz Niederholzer. I'm a tree crop farm advisor with UC Extension, and I'm also the research coordinator at the Nichols Soils Lab, which is a private research facility that basically hosts public research. Very strongly supported by the Almond Board for many years. And the information I'm presenting today is available also with the poster downstairs. So a very quick run through. This is the background on Nichols. It's in Arbuckle, 45 minutes north of Davis. Class 2 or Class 3 gravelly loam's not a high vigor site, but a very good site for almond growing on light with micro irrigation. Mr. Nichols provided it, a formal arrangement with A&R, so I get to work there and on my job description. And you can see the acreage, and we appreciate the Almond Board support because research facilities are not as efficient as farms, and we're supported by crop sales, and you all know the story there. So we've had tremendous support from the Almond Board and the ag community. Projects that I'm going to report on today, there's a dozen or more research projects at Nichols, but the ones that are directly supported by the Almond Board that we report on: organic almond production demonstration block planted by my predecessor, John Edstrom, back in 2006. It's a level rootstock, which nobody plants anymore, but double line drip, buried, just to see how organic production systems would work. And there's a comparison with a conventional block. Same orchard planting, just different tools used different places. And we also looked at comparison of high-value non-pareil pollinizer planting to a self-fertile almond, happened to be Independence. This was planted in 2013 when Independence just began to show up on the Almond Board's reporting. 15 by 20 on Viking rootstock, double line drip. And then finally, we're basically reproducing Rogers' spacing trial that was done out on the east side of Stanislaus County, planted in 2000. Ours was planted in 2017. You can see the spacing, looking at different spacing down the row on 21-foot row spacing between rows on Titan or Peach Almond Hybrid or Rootpack R, again with double line drip. This is Nichols is gravelly loam over heavy clay soil, so drip is we get a lot of runoff on the rolling terrain. Drip is a good way to approach it. Let's see. There we go. This is the organic production demonstration. Organics on the left. The idea was to basically demonstrate, to look at certified organic production practices and materials and compare with conventional. Could you make a go of it? How the trees look, what's yield like? It's a demonstration block. It's not replicated, but there's there's 5 acres of the organic and 2.5, 3 acres of the conventional. You get a good look at production over the orchard, over that space. This is 17 years of yield data comparing conventional. The dark line is conventional, annual conventional production. The light line is, the orange line is organic production. You can see it ranges from over most of the years, 60 to 80%. The conventional production was 60 to 80% of the organic. Once we figured out rust on the left-hand side, let's see if I can get the slide. Here we go. This is a percentage of organic production over conventional. So the higher that the blue line, the better the organic production relative to conventional. And when we had leaf rust problems, we had quite a bit of leaf loss and poor yield. Once we, thanks to Brent Holtz, we were using sulfur for rust control, liquid organic nitrogen fertilizer, and some pelletized feather meal, and we're much better production relative to conventional. And we felt low, as we moved away, having some equipment problems, getting to lower leaf nitrogen in the organic, and our yields have decreased relative to the conventional. Comparing self-fertile to non-perennial and perennials, pollinizer systems, the 100% numbers here indicate solid independence planting. It's 5 rows by, I think there's 20 or 25 trees in a row. Pretty good, for an experiment, pretty good size spacing. And then you can see the non-perel is it's 25% Sonora, 25% Aldrich, and 50% non-perel. It's a 15 by 20 foot on Viking, on not the most vigorous ground, and you can see that the Sonora and non-perel fill their space to some degree. They're about 60, 70%, well, 60 to 65% light interception, whereas the independence, the growth is good. The yield is on a per light basis strong, but they're just not the same size trees. So it's hard to, as Roger was talking about, if you plant everything the same, but you have different sized trees, it's going to skew your yield numbers. But for us here, the most important message was the consistency of production, comparing production over time. This is, non-perennial and Aldrich generally produce more than independence in this planting, but they're bigger trees. Independence yields are consistent and equal to non-perennial. The blue line is independence. It's been consistent up until the last year. We got 3 inches of rain within a few days right at independence bloom, and I think that explains the drop in yield all over, but particularly that the yield drop in independence. The wild card in this particular comparison has been Sonora. It's either been great yields or terrible yields, and that's basically the spikes and the ups and downs in the pollinizer non-perennial system mirror bloom weather conditions. Independence has been much more consistent over time, with a couple of those lowest independence production years have been miserable cold wet, extremely miserable cold wet bloom conditions. Otherwise, their production is fairly consistent. And you can see, I won't read it to you. So finally, we're looking at a tree spacing trial, 21, 14, 16, or 18 feet down the row. They're all 21-foot row spacing. We looked at Titan or root pack R, and we broke the block out because in the middle of the orchard there where the root pack R, there's some heavier ground and root pack's got plum in it, so we thought we'd preferentially put the plum there. And we had 2 acres on the very right-hand side of what you're looking at. We put in K86. But look, those are our varieties all over the same varieties from one end of the orchard to the other. This is just what they look like as a couple of years ago, but they relatively similar light interception across, at this time, across all the blocks. Cumulative yield depends upon the variety. There is some difference on the Titan, very vigorous rootstock. Some increased yield on the non-perennial compared to, we'll look at say 12 to 14 feet for the tighter spacing versus the wider spacing. Actually, the significant difference statistically comes at the 14 versus the 18. The bottom line is, cumulatively, there's some increase in yield for nonpareil over the wider spacing. You don't see it for Aldrich or Kester. And you also don't see it every year. The data you can see, this is 12-foot spacing every year at the different spacings, and they're not all that different. No one's going to go broke at the wider spacing, but there is a little bit of a yield bump in the nonpareil. And Roger's work shows that the tighter trees are smaller over time, easier to shake, less trunk damage, and the real difference to this trial may show up as it did in Rogers in the second decade of the planting. But it's nice to see that super tight plantings, relatively tight plantings for on a vigorous rootstock, on a peach almond hybrid at 12 feet, that's pretty tight. Your yields are still good, and the tree health is good as well. So all this information is downstairs at the poster. Really appreciate the support from Almond Board as well as the entire community, and thanks very much.
All right, we only have 3 minutes left, and I know some of the speakers had to rush a little bit. So is there any message, key message you want to share as a speaker that you didn't have time to share as a closing remarks? If not, I had a question for Mohammad about whole rot. You mentioned there's a toxin that doesn't only kill the spur where the pathogen enter, but also a little bit of the shoot going backwards. That toxin, of course, is very significant because it will kill not the bearing spurs, but the spurs that are non-bearing, and then it could have consequences for the future years. Is that toxin always, fungal dependent, or do you see it across the board in Monilia, Rhizospora, or the other pathogens? Or it's only for one of the pathogens?
So we mainly understand that this is in the Rhizopus, the fumaric acid, but we have not investigated the Aspergillus niger. I have a theory about what might be a different acid that might be produced by the Aspergillus, but we have not worked on that yet.
Okay, thank you. And then for Franz, maybe to clarify the total yield per acre. I mean, in the plot where you show independence versus nonpareil plus the pollinizers, yes. When you put everything together per acre basis, so nonpareil plus the pollinizers versus independence, which one had higher yield? Because you show Sonora had up and downs, but so that will, and you show nonpareil had high yields, but then it's a combination where is the apples to apples comparison. So could you explain that a little bit better?
Okay, let's see. Cumulative yield, the further out you go, of course, the differences are going to be stronger because of the size difference between the trees and the number of good years that'll be in there. This is our last year, was our 13th leaf. We are seeing significant differences, higher levels of production in the nonpareil over the independence, based on the tree size. Income levels, if you use the industry, we use our processor values for each year going back, which just after 13 years, we're just beginning to see a difference in income levels between the two treatments. But for many, many years, incomes were not significantly different. And as I say, they were steadier. For many of those, the independence production has been more steady over time, with the exception of this last year, which I said was a tough, the early bloom got wet.
Okay.
I hope I answered your question.
Yeah, I mean, 1 acre of nonpareil plus Sonora and Aldrich versus 1 acre of independence.
Yeah.
The overall yield, not by variety, but this acre versus this acre.
Because it's a good question. I haven't really looked at it because of the price difference. I just didn't want to put a single pound number on because I thought we were looking really at price.
Maybe something we can look up.
Yeah. And the one thing that take homes for me is anybody shows you yield difference for 1 year or between two treatments, it depends upon the year. As you can see, we bounced around.
Yeah, okay. Well, thank you. Well, that summarizes this session. Thank you for coming. We have lunch next, and then all the speakers are going to be, these speakers plus many more speakers and researchers are going to be in the poster session. So please go to the poster session. It's really a unique opportunity to have all the researchers sitting down, sharing what they do. So try to pick their brain as much as you can. Thank you.